last presentation by Dr. Heba Mostafa at Johns Hopkins. Heba.
Hi, everyone. Thank you, Carlos, for the invitation. And I'm going to just talk very quickly about our data from 2022, our enterovirus D68 surveillance data. We were actually interested in tracking enteroviruses in general in my lab. And we were at the same time actually running the surveillance for SARS-CoV-2 and also influenza. So we were tracking the positivity of the respiratory viruses in general. And I was working in collaboration with Dr. Eric Klein, who also was feeding to us the clinical data from the hospital about any increases in influenza-like illness. So you can see here, this is our positivity data for respiratory viruses from our lab, and this is from the whole Johns Hopkins health system. And you can see that like during the time when we had a lot of COVID, once we started to relax the infection control measures, we had that peak of enterovirus, rhinovirus. This was in 2021, and we actually published this data because we had D68 circulating at high rates at that time frame. But then same thing actually happened in the time frame between August and October of 2022. This is actually the time frame. I will just quickly show the data for now. And this data was actually also just published in the Journal of Clinical Virology. So this is how Dr. Eric Klein pulls our clinical data. And this is again our positivity data from the clinical lab. So in the time frame after August 2022, where the COVID encounters were low, we started to see that increase in influenza-like illness in the at Johns Hopkins health system. And this actually correlated with an increase in enterovirus, rhinovirus positivity rates from the clinical respiratory specimens. So what we did, we actually did, in addition to surveillance from the positive enterovirus, rhinovirus samples, we also collected some influenza, RSV, SARS-CoV-2 negative samples from symptomatic patients. And we collected those specimens to look for enterovirus D68. So we had two different cohorts here. One cohort that had the rhinovirus, enterovirus positive samples. And the second cohort had negative samples that were tested for influenza, RSV, and the SARS-CoV-2 from symptomatic patients. And we were able to identify enterovirus D68 from both cohorts. And we sequenced those D68 specimens, and we were able to recover 75 complete genomes at that time frame and the 32 partial genomes. And we used this data to analyze the genomes for phylogeny. We compared those genomes to the genomes we had from 2021. And also we correlated the data to the clinical data we had. But in general, we noticed that the enterovirus D68 infected patients were largely pediatrics with an average age of 4 years. The majority of those patients required supplemental oxygen. 50% of those patients were admitted, and 27.3% of those patients required intensive care. The genomes from 2022 are in blue here, and some of our genomes from 2021 are in black. So we also saw that those genomes made this cluster within the B3 plate. We actually had some help from Next Plate team who were able actually to share our complete genomes and put it in the tree. And this was actually the same time when the CDC had the health advisory about severe respiratory illness associated with rhinovirus and or enterovirus, including enterovirus D68 from different states. And they, at that time, we were actually in communication because we already had the the data that we had also a lot of enterovirus D68. But we also had in our institution, as I mentioned, enterovirus D68 in 2021 as well. But this year in 2022, the total number of cases were higher. And globally, if you look at our genomes in the tree, it looked like there is a little bit of higher distance of the genomes we characterized in 2022. And we looked in depth to the genomes, and it looked like we had some changes this year in some of the genomes, and at least two of the important antigenic epitopes, B, C, and DE loop changes. We identified some combination of amino acid changes that were not really characterized, at least from the US before, including the 95T and the 98A within the BC loop. We detected those in about 24 genomes. And from the DE loop, we noticed the 143N and 146A from about 7 genomes. And it was actually quite interesting to see that the patients who were infected with the genomes that had those loop changes actually had significantly higher relative viral loads from the clinical samples. However, this data actually we are trying to validate by adding more genomes from that past season. So we are currently still sequencing and adding more clinical data. So this is really a very quick summary for our study from 2022, and just a quick acknowledgement here for my research and the clinical teams. Thank you so much, and I can take questions.
Thank you, Eva, for these interesting observations in Maryland. Is there any question for Eva?
I have a question, Skari, and it's based on my lack of understanding of how to interpret these dendrograms properly. So you showed your dendrogram that was like a little more circular with all of your isolates in there and then comparing to some old, yeah, that one. So are all of these whole genomes, or are, I know yours are, but are all the A's and D's and C's and stuff, are those also whole genome based?
Yes, to the best of my knowledge for this one specific tree, my postdoc actually pulled only complete genomes.
Okay, cool. One thing that so based on the slide from Next Strain that you had, where you guys put the sequences up, I interpreted when those sequences came out, that 2022 cluster is like on the, I guess, y-axis here, almost the exact same as what you see in that 2018 cluster.
Yes, they are very close.
Right. And so my interpretation of that then is that the difference between 2022 and 2018 is actually larger than the difference between, for example, 2018 and 2016 on that graph, because there's more y-axis difference. And so we have the most similar virus we've ever seen over time, and yet clearly the most disparate clinical outcomes, and that we have only respiratory disease. And
correct. Yep.
Okay. I guess that was just a question for clarification to see if I'm thinking of it the right way.
Yeah, I think you're absolutely right.
Okay, thank you. Yeah.
We are trying to put together like a team to also add some serology analysis. We are trying hard, me, Carlos, and a few others from Hopkins to fund the study to look into like the antibody contribution to this phenotype. And I think this is really the missing kind of piece of information here.
Thank you.